It's really about the basic biology of sharks and how they use rollers to understand their behavior and also they can understand ways where we can protect humans or protect sharks through the development of things like shark terror devices. It should provide the good background for Charles' talk. We're talking more detail about some personal fault terrors and other things, which obviously relate to shark behavior. And hopefully, knowing a bit more about sharks and their biology and what fascinating creatures they are to inspire us to find non-lethal ways to reach on sharks. So, I study shark neurobiology. Why is that? Well, they're a particularly fascinating group of animals. They're a very ancient vertebrate lineage that evolved from the main vertebrate stock of 400 years ago. And in that time, they've had ample opportunity to radiate into a very ecological, diverse group of animals, with well over 500 species occupying a range of habitats from freshwater to marine, from coastal to oceanic, and nocturnal or diurnal species tree. And you'll see from just a snapshot here of just 8 species within that 500 odd which exists today, what huge morphological diversity, and that reflects the ecological diversity as well. The other reason to study sharks is they're obviously very important for our ecosystems, as has been mentioned today already. And in many cases, they provide important roles as apex predators in those ecosystems. And if we want to understand more and have better information for their conservation management, very useful to understand how they perceive the world around them, this could help in terms of predicting how they might respond to bionuclear change, or other more applied aspects, such as trying to reduce bycatch by developing technologies like these that can set along lines and prevent sharks from getting caught on the hooks. Obviously, by understanding sensory systems, we can understand more about behavior of the others, and that may help us to design things like shark deterrents. And what I'll do today is talk about scientists where our understanding of the sensory biology is enabled us to think of all ways that we could create a deterrent device. Now, sharks have, just like us, a range of sensory systems. They have some that will be familiar to. They also have some which we possess, unusual but spectacular in their own way. Though they also are hearing, they have specialized sense called the lateral line, the mechanosensory lateral line, which is a sense of dissonant touch. They have visual, smell, taste, they have touch, just like us, and they also have something we don't know, which is electroreception, the ability to detect electric fields around us. Now, one of the most important things to remember about sensory systems is that they predominantly operate over different distances, so different spatial and temporal scales. And this is important for thinking about the information available to a shark and how that is a behavior precision. So, for example, the vision is often used less than a handful of meters, and that's largely because physical properties of water. Water can be very turbid, it scatters light, makes it difficult to be vision over a long way. On the other hand, things like smell and hearing, the sensing which can operate over much larger distances and might attract a shark into an area, for example. Now, what I'm going to do today, just in the interest of time, is focus on 3 sensory systems. That's electroreception, vision, and olfaction. So, sharks have been known at various points in the past as screwing noses, and this is because of their very highly developed sense of smell and their ability to find those from a long way away. And it's easy for this. Sharks will be very sensitive. If we look at the threshold sensitivity, the smallest amount of chemical a shark can detect, it's on the order, to some now, of 10 parts trillion, which is far better than any about modern electronic sensing technology can do. So they're still masters at detecting substances dissolved in the water. And there's an old adage that sharks can detect a drop of blood in an Olympic size swimming pool, which gives you some idea of just how sensitive that olfactory, that smell system is. Now, in order to do this, they have a highly specialized nose, essentially, and it obviously looks very different by our own. And this is the underside of a shark. Here's its mouth. This is a vole. This is a benthic shark that has some, and this is its nostril or an air as its core. And this is where water flows into the nose of the shark and stick a big strength and pulls for the water. And it does that through this very highly folded, pleated structure here called the olfactory rosette. And on that olfactory rosette are a large number of very sensitive receptors which pick up those chemicals. Now, if you look in more detail at the olfactory system of a shark, and here we have a shark, we're looking from the top here, and if you take away the top, the skull, the chondrocranium, you can see the brain sitting here. And these branches to the left and the right are actually the olfactory bulbs, the parts of the brain that deal with smells. And these are very large compared to other, certainly compared to humans, for example. This is just another brain of a different species of shark. And you can see these enlargements with big nerves coming off the front of the brain, highly specialized for smells. Now, I mentioned that there's a specialized organ called the rosette. If we look in detail, it's here. You can see that it has many, many folds in it, and those folded structures are themselves folded again. It's a way of dramatically increasing the surface area to maximize the ability to capture those chemicals in the water that accumulate. And one of the fascinating things about all animals, and sharks are no different, is that there is huge variation between species, which presumably relates to their behavior and their ecology and the tasks they have to do as shells. And this is just a selection of different cartilaginous fishy folk, chimeras, some stingrays here, and some different types of sharks. These are their brains, which we see all very different. And their olfactory structures, labeled in red here, also of very different shapes, with like natural body morphology, and different size compared to the overall size of the brain. And that tells us that the different species of shark smell maybe more or less an off as a sense by raw. And that tells us a lot about the behavior of those animals. Now, if you look even closer still in more detail at those olfactory organs, you can see that they're covered in these filamentous processes called microvilli, which sit on the top of receptor cells, and on each of those are specialized receptors which detect different chemicals, just the same as in our own noses. And what happens is that those chemicals in the water will latch on to the receptors on those cells, and they will create an electrical stimulus, which then travels to the brain. That's how sharp noses detects the smell. Now, again, one of the things we do know, as well as that difference in morphology, if we look at the number of genes that are expressed in the nose between different species, we can see that there's a big difference between different species, again, which likely reflects their ecology. So here we have a mako shark, which has relatively few olfactory genes, olfactory receptors expressed in their noses, whereas some other types of sharks, the benthic species here, Sidorhynchus, has many, many more receptors there. And so there's likely differences depending on where they live or type of food they eat and others of this laboratory as well. Now, one of the amazing things about sharks is their ability to locate smells in the water. And what I'm showing here is a picture of a fascinating experiment where people put tubes into the left and right nostril of a shark, and they put little puffs of smell into each nostril at different times. And what happens is if you put that smell into both sides at the same time, the shark thinks an object is straight ahead. But if you change the timing of the smell coming into the left or right sides, it can tell which direction that's coming from. And I'll show you that hopefully with a cartoon here. So on the left, we had our prey item of interest, our straight ahead of shark, and the odor travels towards that shark in the current and then stimulates the left and right side of the nose at the same time. The shark will swing straight forwards using its directional mechanism. On the other hand, if we have the fish off to one side, there's a time difference and you arrive at smell in either side of the nose. And the shark then knows to turn towards that sense of smell. And the most amazing thing is that they can tell a time difference of a 10.1%. And to give you some context there, an Olympic sprinter's reaction time is a quarter, so 2.5 times slower. The sharks are pretty amazed at finding odors in water. Now, in terms of the development of shark deterrents using olfactory cues, we do have some information that sharks detect their predators using smell. And they do so by detecting what are called semiochemicals, which are chemicals given off by biological peripheral objects in the water. And in a lot of the experiment, some of the shark dislikers will state trachea mobility, would you roll it on their back, and they squirted some water, predator aquarium, so it's an American crocodile, took some water from the crocodile tank, and they squirted it into the nose of the shark with an appropriate control, and the shark woke up and freaked out. And it's a kind of hardwired predator response, potentially, that they're responding to that particular smell. It's also thought that sharks respond to what are called necromodes, which are chemicals given off by then carrying animals. And in fact, people have gone so far as to ferment shark flesh and put it in a can and try and sell this as a shark repellent. There's a couple of problems with that. Firstly, you have to see the shark coming and get the can out and squirt it in the water, which is a bit slow. And the other thing with all chemical deterrents is they disperse very quickly, and so they're not a good long-term solution for scaring sharks away. But knowing something about shark olfaction helps us to consider some of these issues. Okay, electroreception. So this is one of the senses we, of course, don't have. In fact, relatively few vertebrate groups do. But it's an amazing sense for some reasons that you'll see. So if you've ever looked at the business end of a shark, you'll see it's covered in lots of tiny little pores, little holes in the skin. And many of those connected to canals as part of a system called the ampullae of Voronoi. And it's these structures which detect minute electric fields. And what do they use it for? Well, all living animals give off electric fields to a certain extent, including eels as a water, by the way. But generally, sharks are interested in the kind of electric fields which might be given off by prey or mates, members of their own species, for example, which they might then use for communication. It's also possible that the system is so sensitive it can detect the electric fields that are generated in their own bodies as they move through the Earth's magnetic field, so they can use it as a system for navigation. Now, the electroreceptive system is incredibly sensitive. So the kinds of electric fields which are given off by something like fish are in the microvolt range, millions of a volt range. But it turns out that the sensitivity of a shark electroreception system can be as low as 5 billionths of a volt. It's an incredibly sensitive system. And in fact, sharks have circuits in their brain to subtract the electric fields that you create by their own muscles as they move. It's an amazing example of computational in brain. Now, those ampullae of Voronoi are very specialized structures. They're related to the lateral line system, and the pore at the surface of the ski is connected via a long canal which is filled with a conductive jelly spit up the wire. And that carries the electricity down to some sensory receptors at the end, which then create the signal which goes to the brain. Now, what we do know is, again, it's a huge variation between species that likely relates to their ecology and the tasks that that animal has to do. So if we count up the number of pores, those ampullae cores, on the surface of the head, we can see that in something like a white shark, there are around about 800 of those pores, 800 electroreceptors. But in a hammerhead shark, it could be as many as 3,000. And we don't know for sure, we think this is due to the different ways that feed, possibly also to the shape of their head as well. Both of these are pelagic animals that guard in the oval water. But if we look at some venting animals, which live on the otter, we can see in the case of this Jackson shark, a horn shark, and it has something to hum it. Maybe that's because it just feeds on things move very much on the bottom of the sea. But when we look at something like the stingray, which also feel keen on bottom animals, how it can happen. Yeah. We don't really know why there's this variation, which makes it such a fascinating sense. And we're doing a fair bit of work to try and understand this now. And we can do that in a number of ways. We can map the distribution of pores over the surface of the head. We can even use things like MRI, radio imagery, to trace those canals within the head. And of course, we can count the pores on the surface. And when we do that, we find that there's a lot of variation between species. They vary in the number of pores, the size of those pores, the length of the canals, and the canal's orientation. And something we've been trying to look at is whether any of that relates to the strength of the electric fields that call the deterrent shark for coming and taking a bait. In other words, an electronic shock deterrent. Just as with that olfactory system, the electroreceptor system can be used in a directional way for finding prey, and sharks can do this in the dark without the use of fishing and other aspects of their sensory system, like the lateral eye. So if you imagine a fish being off to one side, that there's going to be no time difference because electricity travels almost speed of light, and so there's no time difference between the left and right side of the body. However, the signal will be much stronger here, whereas it will be weaker on the other side, and it then knows to turn and find prey. And one of the other things, are fascinating things about the electroreceptor system is that the sharks can also use this to detect potential predators. So what I'm going to show you here is a video of a shark embryo inside an egg, and you'll see in this video, you'll see it breathe and you'll see it gill it, and then at some point we turn on a very small electric field. You'll see the response. So here's the embryo here. You can hopefully see the gills moving. There's a bit of a countdown to when the electric pulse is going to come on. Comes on now, and you can see that this little embryo stops its gilling. It sits there and waits several seconds before starting gill. And it's thought that this freezing behavior is very similar to other freezing behaviors that we see with animals. They're keeping very still so that they don't give their position away. In this case, so that they don't produce electric fields in the water by moving the gills that a predatory shark or someone else could detect and home in on. Now, this highly sensitive electroreceptor system then lends itself to the question, well, what if we get a really strong electric field? How would they respond to that? There's some very early work done by people who are here to go, decided to see if they use strong electric fields to deter sharks. And this would be, as it's already been mentioned, that Charlie's going to talk a lot more about this, to varying degrees of success, people who've come up with electronic deterrents that create very strong electric fields. And we've also heard about the electric barriers might be used as well. So again, knowing about sensory systems of sharks informs us what we can do to try and turn in a non-lethal way. And just to come to videos here to show just how effective those electronic deterrents can be, here's up a white shark. Now on this board up here is a device that sticks a very strong electric field into the water, and you can see that the shark coming into the bait is deterred quite effectively from taking her bait. One of the things you notice there is the shark got very close. Now other animals seem to be a little bit more susceptible, and this comes back to that idea that the morphology of the electroreceptor system might be a bit different. Is a white-tip reef sharks we've studied. Again, this is where the electric field has been generated, and you can see the shark is twitchy, some big muscle twitches, which don't bond the sharks. and then squirrel away the grey viscers from the ball. Okay, vision. So you can see from this slide here that the eyes of sharks very clearly different, okay, and also very different from our own. And again, this speaks to ecological variation in the design and the sensory systems. So here we have a diagram of our own eyes, and you can see that we had a relatively thin lens and a cornea, which is highly curved. And if you compare that into a shark eye, you can see a couple of things straight away. The cornea is less curved, and the lens is much bigger. This is because when you put cornea in the water, optical surface in the water, the difference in optical power between the water in the lens is very much less. It is between there and the cornea. And so it has to do all of the focusing with this B lens, which is why it moves that physically backwards and forwards from the retina to produce a focus image of the world outside. Now, one of the things that I'll be very interested in is what wavelengths, what colors that a shark was attracted to. We've heard things like young yellow. I'm sure if you're familiar with that term, but the idea that sharks are attracted to particular colors. And I think Mick Fanning swore he was never going to have a yellow surfboard again after being bit by a shark. So I've been looking at the retina of sharks and trying to understand what was going on. And if you look at the retina between a human and a shark, they're actually not too dissimilar. And they have rods, which we use for very dim vision, so at night, when the moon's out, and they have cone photosenses, which are the ones normally responsible for color vision. And if we look at those and then ask the question, well, what wavelength, what part of the visible spectra they're sensitive to, and do they have different types of behind like we do, which might allow you to see color? So if you don't know, humans have three types of cone, and the blue, the green, and a red photoreceptor, red cone photoreceptor. So here's the blue, about 420 nanometers in the blue region spectrum, and they have a green, we have a green and a red CST as well. And that's the color light, of course. Now, you should know that all this from primary school, that mixing those different colors gives us color perceptions. And that's achieved at the level of the retina by comparing the signals from the blue, the green, and the red cones. For example, by comparing the red and the green cones, this gives us a sensation of redness and greenness and so on. So in order to have color vision, then, we need to have at least two of these different cone photoreceptor types. Now, what's quite fascinating is when we look at sharks, they don't have two, all they only have one. So this is a white shark. This is its cone pigment. It pinks in the green file spectrum, here about 500 nanometers. The bull shark is a bit different. It's most sensitive to light in the yellow part of the spectrum of characteristics, used to hanging out in murky water, which tends to be a bit yellow over heterogenic water. Peaks around 5 to get. But both of them have only one coat top in their ready. And what this means is that sharks are completely colorblind. Whereas we would think about people that colorblind, you might be red, green colorblind, but totally colorblind. So if you saw is just shades of gray. And this is relatively unusual across the animal kingdom, including bees, fish, non-shark fish, dogs, cats, and so on. Most animals see color to some degree, but not sharks. It's quite an unusual finding. But the most striking thing is that the ultimate groups to, so basically abandoned color vision, are the marine mammals. So whales, dolphins, and seals, which should come back to the loss from terrestrial ancestors, which had color vision of loss. And so maybe color vision is, if not very useful, for perhaps large annoying predators. And maybe it's even detrimental and may prioritize other visual signals over the cuff. So, in fact, when we look at shark eyes, this may well be the case. So they have a reflective surface that the eye, much like cats do. If you ever caught the eyes of cat, got car inlights, you can see they have this characteristic eye shine, which is a way of capturing more light by reflecting back into the eye for another chance to absorb the light. What else do we know about shark visual systems? Well, we know that their acuity, their ability to see fine detail, is much worse than ours. This is just a cartoon showing the difference between how we see an object and how a typical shark would see an object. Our visual acuity is around about 30 samples per degree, whereas sharks are between 2 and 10 samples per degree. And that inability to see fine detail that is markedly different with humans, and so the world goes less in detail to a shark, certainly at a distance compared to us. Now, knowing these things about the shark visual system, they lack color vision, they're very highly specialized to be sensitive, and they have poor acuity. You can see thinking about this kind of incident and whether the way a shark sees the world can explain some of what happens. And if you think about the idea of the shark's vision is not that good and it might mistake but it's seen for something else, we wanted to explore whether some shark attacks, specifically white sharks, which tends to think of things on the surface, give you examples of misidentity. Now, this was a video just created by a news channel who was doing a story of that sort. And you can see here, someone had like a surfboard and a seal going side by side. We strip out the color information and then we downscale all the image to make it look a bit blurry, which is how shark I see those objects. And you see that they start to look quite similar under those conditions. Now, I won't talk in too much detail about how we did this, but what we did was to compare the appearance of things like seals, surfacing swimmers, to a shark based on what we know about the virtual shark visual system. And these are measurements of the motion cues, the visual motion cues given off by those objects as they move through the water. And you can see that the shape of these, and that's all you really need to sort of basis, are very, very similar. But if you compare those to a control object, just a rectangle here, you can see that the motion cues are very different. And so intuitively that tells us that these objects are all going to look very similar to the shark. And if we look at the shape of the objects as well, there's ways you have shapes of objects in an objective fashion. What we see that someone hailing a surfboard and someone swimming actually to a shark look a lot more similar to a seal with slippers out than to a seal swimming along with this tight teeth. And that's, that's quite striking. I think, these are the same animal and somehow really quite similar to it. So we think that at least some shark attacks might give results. And white sharks are a particularly nice model to study this in. Now, we want to go one step further, and that was the thing about, well, now we have this hypothesis, can we test it? And what we wanted to do is take some seal-shaped photo decoys, which can trigger about reaching the every white sharks, where they path had all the water, and seize this object from the surface, just like they often do with seals swimming near the surface. And we went tow those up and down. We went to a place in South Africa where there's a lot of white sharks flew this. And we saw if we could change their behavior by breaking up the appearance of that object. In this case, by putting lights on the water phase defaults. And just to give you an idea of what those experiments look like, here we've got a rope which is towing this foam decoy, just some little sand eggs here to keep it way typical water. This is attached to a boat, it's trying to roll out a couple of knots. And what you see is this sort of fairly fussy breach guide where the white shark launches out the water and tries to catch what it thinks is a seal. Now, we did many hours of towing these decoys backwards and forwards, and a couple of the results are shown here. We have had control sealed decoys here, and we have so lights in different configurations on those cells, and you can see that the behavior has changed quite dramatically. We've got two behaviors here. We've got that bridge time behavior where they jump out of the water, and we've got what we call follow, which is really where the shark just travels along behind, a bit like Chris showed, following along his UAB, but that often precedes a breach on its targets. So we've got combined behaviors here, but this is the control with their lights, and then we have thin and thick bands of light, so you can see that the sharks just so they rely on. We did actually look to see whether the orientation of the lights made a difference, and it seems they do to some extent. If you have a long stripe that's parallel to the direction of motion, it seems to be less effective. But if you have particular direction motion, it still seems to be very effective. So this nicely summed up this idea that you can go for understanding the very basic biology of shark to trying to understand its behavior, and then try and manipulate that behavior in this case to try and protect surfers.
Where you might get a blast, there's a brutal snap belts.
Okay, that's it from me. I just want to thank all people, many of you are represented here today, just for all their book studies and which I talked about today. And merci.